Unit 8, Molecular Genetics, 8.2, Basic Molecular Genetics and DNA Replication. That's a whole lot to cover in one single video, but we're going to do it. Here we go. DNA, a little bit about DNA and chromosomes. So this is prokaryote on the left, eukaryotic cell on the right. And remember, prokaryotic cells do not have a nucleus, so their DNA is just kind of free floating in the cytoplasm. And the DNA is a single circular chromosome, right? And whereas in eukaryote you have multiple linear chromosomes, and in both you have the chromosome that are going to be kind of found in this substance known as chromatin. And chromatin is just kind of where DNA is just kind of loosely found in the nucleus. It's going to be wrapped up on these proteins called histones, and histones kind of like, think of them as like little spools of DNA, and then when the cell goes to divide, all of that chromatin is condensed into more condensed structures called chromosomes. And so DNA is less condensed chromatin. When the cell divides, it turns into chromosomes. Let's talk a little bit about DNA structure. A couple things. DNA is composed of monomers called nucleotides, and nucleotides are composed of three parts: phosphate, sugar, and base. That sugar in DNA is called deoxyribose, 5-carbon sugar, and the bases are adenine, thymine, cytosine, and guanine. It is found in this double helix structure that you see there on the right. It's a two-stranded molecule, whereas the phosphate and sugar kind of form the backbone, and those nitrogenous bases form the center area, almost like rungs of a ladder. And they have complementary base pairing. So when we say complementary base pairing, what we mean is that G always complements C and A always complements T. You can see that on this picture, right? It doesn't matter which side it's on, but you can know that on the other side is going to be the other one. When you see a G, the other side's going to be C. When you see a T, the other side's going to be A. And that's always going to be the case, at least with DNA. So let's talk a little bit about the difference between DNA and RNA then. You can actually see all three differences on this picture. DNA is double-stranded. RNA is single-stranded. Now, RNA is weird because it will sometimes like fold up on itself and look like it's double-stranded, but it's really not. It's a single-stranded molecule because you only have the one sugar-phosphate backbone. Speaking of sugar, there's a difference in the sugar too. Notice that the sugar-phosphate backbone on the DNA is really dark, where it's a lighter gray on the RNA. It's because RNA is made of the sugar ribose, whereas DNA is made of the sugar deoxyribose. And you've probably already discovered, as far as C, G, and A are concerned, it's all the same. But with T, with thymine in DNA, it is replaced by U or uracil in RNA. So we talked about this in the last video, where A and G are called purines, and C and T are called pyrimidines. U is also a pyrimidine, right? So you don't need to know the difference in shape of them, but you can, because I'm not going to have you recognize the shape of them. You just need to know purines and pyrimidines, those basic two groups of those things. Let's look at the comparison between the function of DNA and the function of RNA. Big difference here. DNA is going to be like a more permanent copy of your genome, right? It's found in the nucleus. So I think of it as like a permanent blueprint. Think of like the architect draws up a blueprint, you spend a lot of money, and it's like a permanent copy. You only have one of them. Whereas the RNA is more of like this disposable copy that goes out on the workplace. It's got scratches all over it, a coffee stain, so forth there on the right, that you see the difference between the two. And so RNA, think more disposable copy. Now RNA has other functions, but we're going to think of it that way. And then DNA is this permanent copy of the DNA that only exists in the nucleus and never leaves the nucleus, just like that blueprint is going to stay in the blueprint house, whatever that's called. There are different kinds of RNA. So the first one is called messenger RNA, or mRNA. This is going to be a temporary copy of a gene. It's going to be transcribed in the nucleus. We'll talk about that. And it's going to go out into the cytoplasm where it's translated into a protein. We'll talk about that as well. Speaking of proteins, tRNA. It's called transfer RNA. The reason it's called transfer RNA is because it transfers amino acids to the ribosome during translation. Big definition. Lots of things going on there. We'll talk more about that in another part. Maybe 8.3. And so transfers the amino acids to the ribosome, so it's called transfer RNA. Speaking of ribosomes, rRNA. Ribosomes are made of two things, proteins and RNA. So rRNA is the RNA that ribosomes are made out of. 3 times RNA. You need to know those. Sounds like a good short answer question. Let's talk about DNA replication. Purpose of DNA replication is to replicate the DNA. Shocking, I know. However, we're going to talk about these 2 other processes that I've mentioned, transcription, translation. And what happens with students is when they hear these other things, they kind of get this weird amalgamation of the 3, like transcriptly replication something. I don't know. They try to make it all one big mess. So just think, if you replicate something, what do you do with it? You make a copy of it. So DNA replication, the purpose of that, make a copy of the DNA. Let's talk about how that works. So the model of DNA replication is called the semi-conservative model. Semi-conservative. Semi means half, conservative means to save something. And so if you look there on the left, you have the original DNA. If you're going to save half of that DNA, well, it splits in half, right? And then each of those halves make a complementary strand. And so the top half there makes a complementary strand, that's blue. It's the bottom half makes a complementary strand, that's also blue. What do we mean by complementary strand? If the top strand is ATCG, then the bottom strand is TAGC. I think I got that right. You get the idea, complementary strand. And so then you have 2 identical strands when you're done. But they're not identical, they're red and blue. Yes, they are, they're identical, right? Because it's replication, that's the purpose of it. So don't get confused. But one of these strands is old and the other strand is new, half new, or half saved, right? So semi-conservative model. Let's talk about some enzymes that are used in DNA replication. First one's called helicase. See there at the bottom. Helicase unzips the helix. That's how I remember it. Helicase unzips the helix. Think about it that way. Primase is next. Primase, you think of like priming a wall, like paint. So it's getting it ready. So primase provides a starting point for DNA replication. Yeah, we're going to keep it there. This is a lot more complicated. You notice there's several things on here that I've erased, and there's a lot more going on here. And this is actually a very simple drawing as well. And so we're going to keep it really simple. So for our purposes, primase provides a starting point for replication. DNA polymerase, it's making the polymer, right? What is the polymer made out of? Nucleotides. And so DNA polymerase is going to come around and it's going to add new nucleotides. Every time it sees an A, it's going to add a T. Every time it sees a C, it's going to add a G, so forth, right? And DNA ligase, think of like ligaments binding things together. So DNA ligase, its purpose is going to be to bind up the sugar phosphate backbone after this process is over, to make, it kind of like cleans everything up. Again, it's more complicated than that, but we're keeping it really, really simple for this class.